Okay then, so I'm going to try and give you a non-technical introduction to the ecosystem modeling package called Mizer. So it's a size spectrum modeling package. So what does that mean? Well, basically it means that we're trying to keep track of how many fish there are of different weights as well as different species. So we're very interested in modeling how fish grow and how their abundances change because they eat each other. So that's really the fundamental idea behind these size spectrum models, is that fish grow because they eat. That means there's a kind of balance in the system, in the sense that whenever a fish grows, it grows because it's eaten something else. So something else has died. And basically, there's a clever way to put all this together in a package so that we can model or at least start to try and model the whole ecosystem and how it changes, how the abundances of different fish at different weights change. It's very exciting. So why is it important to keep track of the weights of different fish? Well, the basic reason is because fish grow over so many orders of magnitude. If you had something like a terrestrial system, it would be okay to say, for example, that lions eat zebra, but zebra don't eat lions. However, in a marine ecosystem, things are a bit more complicated. You might have some kind of fish, say fish species 1, that usually predates on fish species 2. However, since these fish grow over so many orders of magnitude, it may be that sometimes large members of species 2 do eat species number 1. So the basic thing that we're keeping track of in Mizer is a sort of record of the abundance of different fish at different weights. Basically, it's sort of like how many fish have you got at this weight and that weight. Now, if you say, for example, how many fish are there at 1 gram, well, there won't be almost any which are exactly 1 gram. It does make more sense to say how many fish are there, let's say, between 0.9 and 1.1 grams. So we want to be keeping track of a type of density function, which is a kind of curve which, if we add up the area under it between two different weights, we get a type of count of how many fish there are between those weights. So we want to keep track of such things for different species, and we want to see how these things change over time. So I've got here a picture which is showing the abundance of different species at different weights, and you can imagine a picture like this is going to be evolving in time because these fish are eating each other and dying and reproducing and so on. Okay, so I've already spoken about this slide. I'm basically indicating that the basic thing that Mizer is built on is the idea of this sort of predator-prey interaction, where, let's say, a weight W predator encounters a weight WP prey and eats it. What's the result? Well, the weight WP prey vanishes and the predator's new weight will be W plus alpha times WP, where alpha is the efficiency of, it's a sort of fraction of energy which is passed on from prey to predator. Basically, thinking about that interaction and doing some maths, we can derive the Mackendrick-von Foerster equation, which describes how the abundance of individuals at different weights and different times changes. And so this is a partial differential equation specifying a relationship between the rate of change of the abundance with weight and with time. And you see it's in terms of the growth rate and the death rate here. Now, if those were just sort of functions which stayed the same over all time, this wouldn't be such a difficult problem to solve. We could actually write down a solution to this kind of partial differential equation. But things are more complicated, in fact, because to determine, say, the growth rate of an individual, you have to think about what kind of things there are for that individual to eat. How much food does it have? And that depends on the abundance of lots of other species at lots of other weights. But the basic idea behind how feeding is modelled is that we'd have a number called the preferred predator-prey mass ratio. And that basically specifies what fraction, W divided by beta is the sort of preferred weights that a weight W predator would like to eat at. So when we're plotting on a log-log plot like this, this value of beta sort of corresponds to how much further down the size spectrum a predator is eating at. So, okay, that sounds a bit cryptic. Let me try and explain what's going on in this picture. So let's think about these. So we've got the log body mass on the x-axis. And let's think about these fish in the middle, these sort of medium-sized fish. What's happening to them? Well, some of them are getting eaten upon by these predators. They're obviously the type of prey size that these predators prefer. And so some of them are going to be getting killed. And these fish themselves are going to be eating these smaller fish further to the left. And so some of those smaller fish are going to be killed, but the energy from killing them, from eating them, is retained by these medium-sized fish, and some of them are then going to grow to become even larger. And so that's going to cause some of this sort of biomass here to move to the right. And we also have a reproduction effect where we sort of add up over the mature fish and figure out how many new individuals are being made in the system. So as I said, the predator-prey interaction is basically governed by this preferred predator-prey mass ratio. Okay, so here's an example. Let's say we have a predator that weighs 1,000 grams. So I've indicated that by this spot here on this plot. And then this log normal distribution here shows the sort of preference function of such a predator for prey of different sizes. And you see, because its preferred predator-prey mass ratio is 100, it most prefers to eat prey of weight 1,000 divided by 100. Okay, so how do we work out then the growth rate of a particular individual in Mizer? Well, basically, we want to sum up. First thing we want to do is find out how much energy that individual's getting. And to do that, we want to sum up over all the different prey that individual will have. So we sort of do an integral. We add up over all the different prey, add up over their abundance, but we weight it by how much preference the predator has for eating such prey. And we also take account of the sort of volumetric search rate of this individual, this predator. And that tells us how much energy our individual has to use. Now, we also have to take account of the eating rate is limited. So we have a type of functional response curve which limits how much of the food available can actually be eaten. And we have to subtract some of the energy away for the costs of movement and metabolism of our fish, and also for the energy required for reproduction. And all that remains after that can be used for growth. So that gives us the growth rate of our individual. The working out the death rate of an individual is somewhat similar. It's almost like working out the growth rate in reverse. Well, kind of. I mean, okay, let me explain. Death can happen for various reasons in our system. So it could happen because of fishing. It could happen because of other effects, which I've indicated by a question mark. We can add in other types of death into Mizer. But one of the main reasons is going to be predation mortality. And determining the predation mortality is sort of like working out the growth rate in reverse. What I mean is that to work out the growth rate of an individual, you have to sum over its different possible prey and weight by your preference for eating them. To work out your death rate, you have to sum up over all the different possible predators and weight by their preference for eating you. Okay, so this is all described in much more detail in the Mizer vignette. I just want to give a rough outline of what's going on here. We also model the background resources. So the very smallest fish, in order to grow, the smallest fish that we're modeling in a given simulation, in order to grow, they have to have something to eat. And we model how there is plankton, and that's governed by semi-chemostatic dynamics. So it's possible to eat the plankton down and it can get replenished and so on. To work out how much energy is, how much reproduction is occurring, what we do basically is add up over all the fish of a given species and weight by how much energy they're putting into reproduction. And then we basically have to take account of limited reproduction efficiency, and we get how much energy is being put into reproduction. We have to divide that by the egg weight. That gives us how many eggs are being produced. And this is sort of, it acts like a boundary condition in our partial differential equation, sort of telling us how much mass is flowing into our system from the left-hand side, from the smallest weights individuals. And basically the result of all this is that we can get some nice simulations where we can keep track of how the biomass of different individuals, the biomass density of different individuals at different weights changes over time, and all the nice things that we can get from that, like determining what the type of fishing yield ought to be if you're using particular fishing gears and so on.